Max, I want to start with you. When we think about AI and drug discovery and the models that you and ISO Labs are building, for the layperson, what does that actually look like on a day-to-day basis for you and the team?
So what we're doing at Isomorphic Labs, we're building this drug design engine. So you can think of this as a machine which is able to come up with new molecule designs for different disease areas, different indications, and even for different modalities, not just a single modality. And underlying that drug design engine are a whole suite of AI models. These are not just a single model like AlphaFold, but actually we're going to need maybe half a dozen AlphaFold-like breakthroughs that come together into this drug design engine. So these are predictive capabilities, generative AI capabilities, things, for example, like AlphaFold that predict the structure of proteins. Proteins are the fundamental building blocks of life. They form these molecular machines. We need to understand what these proteins look like physically in 3D, and also how these proteins interact with other biomolecules, things like DNA, RNA, small molecules. Small molecules are the class of molecules that are often drugs that you take as pills. So if you can start working out and predicting, okay, what shape is this protein going to take when it interacts with DNA, and if I design this small molecule drug, how is that going to come in and affect the shape of this system? Because ultimately, if you can start changing the structure, 3D structure of this system, you start modulating the function. You change the function of this molecular machine. That changes some functions within the cell, and ultimately that could change the phenotype or the state of your disease. So it's these sort of components like AlphaFold, these structure models that can understand how strongly these molecules bind together, binding affinity models. And then, of course, drug design is not just about making a molecule that will modulate your molecular machine in a particular way, but it also has to be a good drug. It's got to be safe for people. It's got to be absorbed into the gut if you're taking it as a pill. It's got to get to the right part of the body. It's maybe even got to penetrate the cell wall to get into the cell itself. So there's a whole spectrum of models that come together into this drug design engine.
There's so many different variables that you touch on there. So Rebecca, given your biomedical background, how do the systems that Max and his team are building change drug discovery from your perspective versus the legacy systems? What are you and the team able to do with these kind of models, this kind of technology that you wouldn't be able to do with legacy systems?
Well, if you think about the way that we tackle drug design more traditionally, it's very iterative. So you maybe make a slight tweak to your molecule, your drug molecule. Then you've got to go in a lab and you've actually got to make it. That might take a couple of weeks, maybe even a couple of months. You've then got to test it in a system which is approximating, your biological system that you're interested in. Then you get the data back, then you have to iterate again. It's very, very stepwise. And this is why it takes so long and it takes so much time. So what we're doing is lifting that all out of the real world. We're putting it in this virtual world so that you're doing all of this on a computer. So you're designing a molecule, you're testing it through these models, which, thanks to the incredible ML group we have, are in some cases approaching experimental accuracy. So you're doing all this design on the computer, and then you maybe multiple iterations on a computer, and you take the best things and you take those into the lab. So you're skipping steps, and you're getting all the way to something that looks much better for half the time.
And that's where we get the shrinking of the time frame that we're aspiring and hoping to achieve. And Max, you touched on AlphaFold, and many in the industry had assumed that it was almost an impossible problem to solve, which is the protein folding problem. You and the team managed to do that with AlphaFold 2 and then AlphaFold 3, which gives scientists and medical experts the ability to look at how those molecules interact. You talked about the need for other models. What comes beyond AlphaFold 3? What other breakthroughs do you need, or other models do you need beyond AlphaFold 3 to get the kind of breakthroughs that you and Rebecca are aiming for?
Yeah, I think we have much better understanding of this biomolecular world today than we had, even 4 or 5 years ago on a computer with models like AlphaFold 2, AlphaFold 3. But we're not the full way yet. There's still a huge amount of accuracy to be gained on these models, and we're seeing big step changes in accuracy internally with our sort of next versions and iterations of these types of models. And then, as I touched on before, it's not just about understanding the structure of these components, that's really just one part of the problem. You need to understand how strongly these molecules come together. You need to understand how these molecules interact with different parts of the body. A common cause of clinical failure is from toxicity, and a big source of toxicity can be from, for example, how these molecules not just interact with the target protein that you're trying to design for, but interacting with all the other proteins in your body.
Yeah.
And so you can imagine that we need to understand not just how these molecules interact with what we're designing for, but with everything else in the body. That's a big task. And then there's this whole question of actually, how do we design these molecules? Because molecular space is massive. It's something like 10 to the power of 60. That's a huge number. So you can't brute force search that. You can't just like ask these models, hey, how does this molecule look? How does that molecule look? People try to do that. You can maybe feed, 100,000, 1 million, 1 billion molecules through these systems and start reading the answers. But that barely scratches the surface of this massive molecular space. And so here, there's a huge opportunity for generative models, search processes and agents to be, instead of having to exhaustively search molecular space, molecule by molecule, have generative models that come up with designs that can effectively search the entire space and triage that down to just, ultimately a handful of molecules that then, as Rebecca said, we can take into the lab and test.
And Rebecca, on that point, what is the biggest challenge in getting, and you were talking about this, the in silico molecules that the models may suggest, and making sure that the in silico versions, the computer versions, essentially work in the real world in the lab conditions? What are the biggest challenges around that part?
I mean, we can think of the models as enriching what we're doing. So they might not get it perfectly right every single time, and we have to be aware of that. We have to have a strong hypothesis for why we're making a particular molecular change. And then once we have that hypothesis and we've got some conviction, we take it into the lab, we make it, and we see. And sometimes we're absolutely blown away by what we get back. We've generated something that's novel, it's potent, it's got the right kind of properties, solubility, and everything else that it needs. And sometimes things don't work as expected. And we would imagine that over time, as the models get better and better and our processes get better and better, we should start to see this increased success.
And what is that progress looking like right now? I mean, do you have days where you kind of stand back and, wow, this progress has been phenomenal? Or are there more days where you've got your head in your hands thinking, it's not working out the way I hoped?
There have been some days where things are just phenomenal. And I'll maybe go and find Max and just say, like, can you believe this? One of our structure models predicting the pose of a molecule binding to a protein always perfectly, and you get an X-ray structure, and it confirms, to just this incredible accuracy, and it's something that's never been seen before. Just absolutely mind-blowing.
And do you and your team now, Rebecca, have a list of molecules that you've identified that could be candidates? Are we at that stage yet?
With some of our more advanced programs, we are moving through preclinical development. And then we have a whole load of other programs coming through that are much earlier, with lots of opportunity associated with those as well. So, yeah.
And Max, on the question of data, because medical data, biometric data, it can be, or biological data, it can be complicated, it can be messy, it can be siloed, it can be biased. How do you ensure that you're getting the data that you need into your models?
Yeah, machine learning, the fuel for machine learning is data. And there's the old saying, garbage in, garbage out. So the data has to be good, and it's got to be the right distribution to learn from. We have a very, very comprehensive data strategy where we will look at as many sources of data as possible. There's a huge amount of publicly available data that works really well. There's a lot of historical data that's also being collected. But as you touched on, it can be highly biased because it's not being collected for machine learning. It's being collected as the byproduct of maybe historical drug design campaigns. So I still believe there's a huge opportunity to actually be creating the data that's needed for these machine learning models. And we do a large amount of work in that respect as well. So actually generating our own wet lab data for the sole purpose of training these models. And
you're confident in the quality of your data at this point?
Yes, I think that's where we invest a lot in, understanding what data is out there, working out how to ingest and clean and really work out where the signal is from that data. These are sort of big areas of research for us, and that's where you can see huge benefits.
And both you and Demis have talked about the generalizable technology that you and Iso Labs are building, which is different to some of your competitors. Just explain why that is important, why that's the route that you've taken.
Yeah, this notion of generalizability, that's the key for what we're building, because ultimately we don't want to do just a single drug design program and then we're done. No, we want to be building this drug design engine, this machine that we can apply again and again on any target in any disease area, even against any different modality. So it's really important. What does generalizability mean? That we have these models that they're trained on a certain set of data, but then they can be applied on something completely novel. They can be applied to a protein, a target that's never been worked on before. They can discover chemical matter that's never been seen in humanity before. And in practice, we're seeing that bear out day to day. We have benchmarks internally that measure how generalizable our models are. But the proof is in the pudding. Yeah. Can you actually use these models on completely novel drug design campaigns?
Is it fair to say that that generalizable model is more ambitious and more challenging? The outcomes can be that much more significant, but the building of that model versus a more targeted one is more ambitious, is more challenging?
Yes, I think that's the case. We're focused a lot on new molecules for new targets, not just changing existing published drugs by small amounts. So we want to be creating novel chemical matter against problems that people have been working on for a huge amount of time but made no traction. So it's really important that we have these generalizable models that can work for that, but they are harder to build. It's much easier to collect a small amount of data around the very restricted problem set that you know about, create a small model around that can be used just in that little restricted problem set, but can't be applied to the next program or to the next program. For us, that won't cut it, that we won't get this sort of reusable engine. And so from day one, we've been building these big foundation models, things like AlphaFold 3, which do strongly generalize to completely unseen systems. And again, we're seeing that across the board of our model space.
And Rebecca, why does it make sense for the internal pipeline to focus on immunology and oncology, so cancers? What was the rationale behind that, and what does that mean in terms of what the achievements could be?
It makes sense to focus on those two disease areas. Clinical trials are much more tractable. We can run them in a shorter time frame. They're diseases with a lot of impact as well. I mean, we all know someone affected by cancer. Immunology is also important as well. It affects a huge proportion of the world, people who have inflammatory diseases. So they made sense as places for us to start. Really good preclinical models as well, with good translatability into the clinic. So you can make a prediction about something working based on models that you apply before you go into humans, and things should translate better.
What is your... Look, I know this is a difficult question because there's all sorts of variables, but given what you see right now and what you're working on, what would be your best assessment as to when we may be able to cure cancer?
I think cure cancer is a massive statement because actually cancer is a collection of a huge number of diseases. But I think we can see this as a stepwise process towards taking people with different cancers and giving them, hugely better outlook, where actually cancer becomes more of a chronic disease, where you maybe take medicine for life, but you have a normal lifespan. And we already see this in some cancers where medical treatments can, give you a normal lifespan. And I think, for me, that would be incredible, right? Cancer is no longer something you need to worry about. It's just something that you need to make sure you get the right treatment, and you maybe have to take treatment, but you have a normal lifespan.
And that proposition is not too far off. We're talking years rather than decades, potentially.
I think it's really difficult to put a time stamp on that. I mean, there are some cancers that are incredibly complex, and especially as cancers become more and more advanced, they become more difficult to treat. So, but I think we can start to think about how we might approach this problem now.
Max, give us a sense of the power of the models that you're using right now. Can they analyse an entire family of proteins, for example? I'm just getting a sense of what they're able to do in the here and now.
Yeah, so these models, they can be applied to not just a single type of protein or a single protein, but we can apply this across what we call the whole human proteome, the whole universe of proteins that are within us. And that means the other thing is because these models run really quickly, the neural network models, we can run thousands and thousands of calls of these models in parallel, which means we can think about analyzing not just a protein in isolation, but whole families of proteins, whole universes of proteins. And this is why this whole world of in silico drug design is such a departure from the experimental world, because in the experimental world, to study a protein, you have to go and synthesize your protein. You have to get it shipped to you. You then have to do the physical experiments. You can't be analyzing thousands of proteins in parallel against the same thing. So it really changes sort of experimental design based on these models.
Is this, you're an expert in you're an expert in many things to do with AI, but you're an expert in reinforcement learning. Is that still the right approach to try and refine this technology?
Reinforcement learning is one of these fundamental tools in the machine learning and AI toolbox. And it's part of the spectrum of tools that we use at Isomorphic Labs. Where reinforcement comes into play, reinforcement learning comes into play, is where, for example, we have very powerful generative models. We have these really powerful scoring systems that can tell you how good or bad a molecule is. And then you want to work out a process of iterating on that molecule and making it better and better towards the sort of design criteria that Becky and team will come up with. And so that's where reinforcement learning can come in, can help train these agents that are able to explore molecular space towards these design criteria for drugs.
On the partnerships, Becky, with Novartis and Eli Lilly, can you give us some detail in terms of some of the milestones that you and the team have been able to achieve in those partnerships?
So these are incredibly collaborative partnerships. I've been working as part of one of them. We work really closely with Novartis. I think Novartis came out initially and said these are really challenging targets. They're targets we've been working on for many years in some cases. Some of them are sort of a graveyard of pharma effort, and we've managed to make some really good progress on some of them. Some of them represent what we might call dark protein matter, where there's never been a molecule identified to bind to these proteins. And we've managed to identify some of the first chemical matter binding to these proteins. So, they've seen some sort of incredible stepping stones from the Isomorphics platform already.
Okay, that's huge. And if you were to, Becky, if you were to look out 10 years, if you project out 10 years, how does this technology, how do you think the balance of power shifts around big pharma versus biotech and some of the startups and some of the tech players?
I think that's maybe quite difficult to predict, but what we see from Novartis is this really forward-thinking mentality where they have really deep scientific expertise internally. Incredible, but they're also really forward-looking, and they want to partner with biotechs who are, really on the cutting edge of pioneering these new deep learning models, so that they can ensure they're using the latest capabilities in drug design to really sort of push that frontier forward.
And Max, Demis forecast when I spoke to him that in 2 to 3 years' time, potentially, we could be at the point where we're reducing the AI drug discovery part of the process down to maybe from years, possibly to months, possibly to a shorter timescale. What are the technical hurdles that you and the team need to overcome to get there?
Yeah, absolutely. This is the goal, and we're really starting to see some incredible progress towards that. The key technical hurdles around this is really being able to, predict more and more about the outcome of experiments in silico on a computer. And so the closer we get our models to that experimental level accuracy, you can imagine the less you have to rely on going out into the lab, synthesizing your molecules, actually doing this manual work. And so really for us, a big goal for a lot of our modeling capabilities is this experimental level accuracy. For some of these, we've actually achieved these and starting to achieve these, especially in different pockets of the chemical universe. So that's one of the real key things for us.
And Rebecca, I'm going to ask you to forecast again, but in 10 years' time, given what you're seeing, given what you and Max are working on, what does our world of medicine look like for people interacting with medicine, interacting with their doctors, having to take therapeutics? How different is it going to look in 10 years?
I think it is difficult to put a time frame on it, but I can see this really exciting future where you don't have to worry. You've got symptoms, you might go to your GP, or maybe you're actually interacting with, some sort of AI tool that can help to diagnose you at that point. And there would hopefully be a medicine to treat your disease. That's obviously way into the future, and there's lots to solve before then. Isomorphic Labs, we'll want to collaborate with others to kind of achieve this big vision that we have to solve all disease. It's a massive ambition, but I think it's a really exciting future where we can just really make a big impact on human health and disease. That's what gets me out of bed in the morning, is really thinking about how can I impact patients' lives with the work that we do.
And solving all disease, guys, does that happen whilst you're both still at Isomorphic Labs? Is that feasible to think that you could meet that goal potentially in both of your lifetimes?
I mean, this is a big ambition, right? There's no denying that. It's also not going to happen overnight. And it's also not going to happen alone. We're going to have to galvanize, not just ourselves, but I think the whole industry, many industries together, to start stepping towards this. What is true is that there is this fundamental new technology, AI, which is quite different. It's generalizable. It can be reused across different spaces. And that's something that's very rare to find more broadly in biotech. And we haven't really seen that happen often in the past. And so we really have an opportunity now to actually step towards, this holy grail of solving all disease.
Rebecca?
Just to echo all this, it's an incredibly exciting opportunity. I think we were talking before that I've worked in the space for a while, and now to be able to use some of the tech and the deep learning coming out of Max's team to actually design molecules in this virtual world, actually make those and see the incredible jumps that we can make in condensing this process down with the aim of getting molecules to patients faster. It's a really exciting time. I've been blown away by some of the technology, and I hope that we get to see some big changes in the time that we're both at ISO Labs.